So, welcome to this class on Neuroscience of Human Movement. In this class, we will be reviewing our discussion on action potential, and I will be discussing a new topic which is basically neurotransmitters. So, in this class, we will discuss the differences between action potential and graded potential, and we will discuss the concept of a synapse, and we will discuss neurotransmitters, and we will discuss how acetylcholine is synthesized. We will discuss excitatory postsynaptic potentials and inhibitory postsynaptic potentials, and we will discuss temporal and spatial summation that we introduced in the previous class. Okay. So, let us remember, a stimulus when it is arriving, it may or may not cause an action potential. If the stimulus is strong enough to take the membrane potential to threshold, an action potential will be caused. Any other situation or any other stimulation that leads to a subthreshold response, or basically a subthreshold stimulus causes what are called as graded potentials, like these. A suprathreshold stimulus by definition always gives a stereotypical transient change in membrane potential, right, which is what we call as action potential. This we have seen. What are the differences between this graded potential and action potential? Let us try and summarize this. Right. Graded potential, depending on the stimulus, may or may not be depolarizing. We saw in the previous class that sometimes graded potentials can be hyperpolarizing, whereas by definition action potentials always lead to depolarization of the membrane. By definition, graded potentials, the response is proportional to the strength of the stimulus, or in other words, the larger the stimulus, the larger will be the response. But action potentials are stereotypical. Once the threshold is crossed, the response is always the same. So, you are going to have a stereotypical response, that is all or none. Either you have an action potential or you do not have an action potential. So, you do not have a situation where you have so much percentage of an action potential. Either you have an action potential or you do not have an action potential, right. And then the question is, how is the strength of the stimulus encoded? We said that the strength of the stimulus is coded in the frequency of the action potential, how frequently it happens, or the number of impulses per second that it happens, right. And in general, amplitude of the action potential is relatively large, whereas the amplitude of the graded potential is relatively small, right. And the duration of action potential is relatively short, for a brief amount of time it happens, whereas graded potentials can remain at a relatively low value for a long period of time. Sometimes the duration may be as high as some seconds, right. And the stimulus driven ion channels are responsible for graded potentials, right. What are these stimuli? These stimuli are ligand gated ion channels, mechanosensitive ion channels, temperature sensitive ion channels, vibration sensitive ion channels, etc., right. So, these are the stimuli that cause graded potentials, whereas action potential is almost always, right, always caused by voltage gated sodium and voltage gated potassium channels, and it is the interplay between their conductances that cause action potential. We have seen this in the previous class, right. And in action potential, there is a period during which you cannot cause one more action potential, right. This period is called as a refractory period. Why is this happening? Due to the inactivation gate of the voltage gated sodium channel that is closing, right. There is no refractory period that is associated with graded potentials, right. And also what you have with graded potentials is that over time it is possible for graded potentials to be summed. We call this as temporal summation, and across space graded potentials can also sum, right. This is called as spatial summation, whereas as soon as one action potential is caused, you have all or none phenomenon. Basically you have an action potential. Soon after that, if another stimulus is caused, it cannot cause an action potential. Why? Because there is a refractory period, because the inactivation gate of the voltage gated sodium channel is closed, the membrane is in a refractory period. So, two action potentials cannot sum. However, multiple graded potentials can sum and produce an action potential, but an action potential itself cannot sum with another action potential even if it arrives at the same time, even if there is a suprathreshold stimulus that is maintained, it cannot cause a higher amplitude action potential, right. So, action potential usually refers to a stereotypical response, right. So, summation is not possible, and graded potentials travel by passive spread or diffusion of ions from one point to another. Action potential travels by regeneration of a new action potential. We discussed this case, right. So, suppose this is the membrane and there is one voltage gated channel and there is one other voltage gated channel. Here there is an action potential, here another action potential is regenerated because threshold is crossed at that point, right. So, this we saw, right. So, at each point in time and at each point in the space, or at each voltage gated sodium channel, a new action potential is regenerated, right. Because there is passive spread in graded potentials, amplitude will diminish as the distance increases. This is to be expected because there is diffusion, right. So, some sodium ions will miss, right. So, not all the sodium ions will travel and reach the next channel. So, amplitude diminishes as the distance increases. So, there is decremental, whereas in action potentials amplitude does not diminish. Why? Because a new action potential is generated. Actually, in practice the amount of sodium that enters at one point reduces, diminishes, but at the new voltage gated sodium channel, the amount of sodium that reaches the new voltage gated sodium channel is sufficient to cause an action potential there, which is why you actually have the amplitude not reducing, the situation where the amplitude is not reducing, okay. And graded potentials are usually brought about by external stimuli. This may be sensory stimuli such as touch or such as pain, or by neurotransmitters released in synapses, and these cause graded potentials, and let us remember these graded potentials can be either excitatory or inhibitory, or they can either depolarize or hyperpolarize, right. Whereas action potentials are triggered by the voltage gated sodium channels reaching threshold, right. So, what causes the voltage gated sodium channel to reach threshold? That may be graded potentials. Graded potentials possibly cause the initial trigger of action potential. After that, action potential travels from one point to another, or in other words new action potentials are generated along the direction of propagation, right. Graded potentials can essentially happen in any place along the plasma membrane. Wherever there are stimulus driven channels that are available, there graded potentials can happen, whereas action potential happens only in regions where there is sufficiently large number of voltage gated sodium and potassium channels, because it is these channels and their conductances that determine the characteristic of action potentials, right. So, these are the differences between action potentials and graded potential, right. So, then the next question is how action potential causes communication with another cell, right. This happens in a synapse. A synapse is a specialized region where one neuron communicates with another, and there are essentially two types of synapses. One is an electrical synapse. Here there is an opening between one cell and another cell, and communication is through ions, right. Virtually there is no synaptic delay. These are called as gap junction mediated communication, right, and usually this communication is bidirectional. So, if there is one neuron here and that communicates with another neuron there through electrical synapse, in that case communication can happen in this direction and also in this direction. So, both cells can talk with each other, in a bidirectional manner. Usually the chemical synapses are more in number. So, usually this is not, the electrical synapse is not the manner in which communication happens from one cell to another. Usually the agent of communication is a chemical called as a neurotransmitter, right. Because there is chemical communication from one cell to another, usually it comes with a synaptic delay, and one cell can talk to another cell. So, there is a speaker cell and there is a listener cell. Usually this is unidirectional communication. For example, an action potential here causes these chemicals to be released, and these chemicals are detected by these receptors, and once these receptors receive these chemicals, they cause an action potential in this region in the postsynaptic cell. In general, when we say synapse, we refer to a chemical synapse, and a neurotransmitter is usually involved. In general, synapses can be classified into multiple types: axo-secretory, axo-axonic, axo-dendritic, axo-extracellular, and axosomatic, depending on what the destination is. If the terminal is secreting into the bloodstream, it is called axo-secretory. If it is into another axon, that is called as axo-axonic. If it is into a dendrite, it is called as axo-dendritic, etc. Depending on the destination cell or the destination, the synapses are classified, or the chemical synapses are classified. Okay. What are the neurotransmitters, or what are the chemicals that we are talking about, right? So, basically from a presynaptic cell, this chemical gets transported by diffusion to the postsynaptic cell, where it is detected and a response is generated, right. And these chemicals are synthesized and released in the presynaptic cell. Usually neurotransmitters are classified into various categories. These are biogenic amines, example is dopamine, amino acids, example is GABA or glutamate, neuropeptides, example is substance P or somatostatin, right. And then there are other neurotransmitters. An important neurotransmitter is classified as other neurotransmitters, that is acetylcholine. It is an important neurotransmitter that is classified as other neurotransmitters. In general, depending upon whether the neurotransmitter is causing excitation or inhibition in the postsynaptic membrane, they are classified as excitatory neurotransmitters or inhibitory neurotransmitters. Examples of excitatory neurotransmitters are acetylcholine, glutamate, and the famous example for inhibitory neurotransmitters is GABA. In general, acetylcholine causes an excitation of the postsynaptic membrane, and in general GABA causes an inhibition of the postsynaptic membrane. However, there are exceptions to this rule. Let us discuss the case of acetylcholine, how acetylcholine is synthesized. Acetylcholine is synthesized by the reaction of acetyl coenzyme A, or also called as acetyl CoA, and choline. Basically, this is made by the protein choline acetyl transferase. This is the protein that is responsible for generation, and it is stored into bags, into small bags called vesicles, right. This stored acetylcholine is released on stimulation when an action potential arrives in this region, right. This detail we will discuss in future classes, right. So, suppose acetylcholine is released from a vesicle, the end plate at the motor side or the motor end plate generates a small potential called as a end plate potential, right. So, whenever acetylcholine is released into the synaptic cleft, a small amount of potential is generated on the motor end plate. This is called as a motor end plate potential, right. The smallest amount of end plate potential that can be generated is due to one acetylcholine molecule, is it not? This is called as a miniature end plate potential. Okay. Now, if acetylcholine is present, suppose now let us suppose this is the pre-synaptic membrane and this is the post-synaptic cell. This is the pre-synaptic cell, right. Suppose acetylcholine is released here and it is remaining here itself, then it could spontaneously trigger new action potentials in the postsynaptic cell, right. To avoid that possibility, chemicals are generated and these chemicals can degrade acetylcholine. These chemicals that can degrade acetylcholine are called as esterases, or in this case, in the case of acetylcholine, this is called as acetylcholine esterase. This acetylcholine esterase basically breaks acetylcholine into choline and acetate. This choline can be reuptaken by the presynaptic cell for making more acetylcholine. Okay. So, choline is reuptaken or recycled, then the question is where acetyl coenzyme A comes from. That comes from the presynaptic cell. Okay. So, approximately 50% of choline is returned to the presynaptic terminal and the remaining choline is excreted, is cleaned out. Okay. Now, what could happen at the postsynaptic cell, right? If multiple excitations come one after the other in time, right, one after the other, this causes an excitatory postsynaptic potential, and another excitatory stimulus causes another excitatory postsynaptic potential. Suppose following that you have an inhibitory stimulus that causes an inhibitory postsynaptic potential. So, if the postsynaptic potential causes an excitation, right, that is called as an excitatory postsynaptic potential. If it is inhibitory, it is called as an inhibitory postsynaptic potential. It is possible for these to add in time, and when the postsynaptic cell reaches threshold, an action potential is generated in the postsynaptic cell. Until then, you only have local currents or graded potentials. Okay. And also let us remember that multiple inputs arrive at the same point in time, right, that is called as spatial summation. Whereas, if you have a situation where one input is happening at a relatively high frequency, or one input train is followed by another input train closely in time, that leads to a situation where inputs can be added or the responses can be added in time, leading to temporal summation. Spatial summation is the situation when inputs arrive at the same time from multiple different points in space, right. If inputs arrive at different points in time, but closely followed by each other from the same point in space or from the same input neuron, that is called as spatial summation. Actually, the output that happens can be a combination of the two. It need not be completely spatial summation or temporal summation. Usually it can be a combination of both spatial summation and temporal summation. From the viewpoint of the postsynaptic cell, as long as the threshold is reached, you are going to have an output, you are going to have an action potential. Okay. So, in summary, we have discussed the differences between graded potentials and action potential. Graded potentials propagates by passive spread, whereas action potential propagates by saltatory conduction or regeneration of a new action potential in the voltage gated channels. And synapses are communication zone between one neuron and another neuron, and there are two types of synapses, electrical synapses and chemical synapses. The most common type is the chemical synapse, and there are different neurotransmitters, basically biogenic amines like dopamine, amino acids like GABA, neuropeptides like substance P, and other neurotransmitters such as acetylcholine. And we saw that this could cause either an excitation or an inhibition in the postsynaptic cell, thereby causing an excitatory postsynaptic potential or inhibitory postsynaptic potential, right. And we also discussed how acetylcholine is synthesized and how it is degraded. Then we also saw spatial and temporal summation. So, with this we come to the end of this lecture. Thank you very much for your attention.